recombinant DNA technology. How we apply biosafety in recombinant DNA technology. Keep in mind that recombinant DNA technology is also known as genetic engineering, in which we combine DNA from different sources. So these are the transgenic organisms that never exist in the nature before. So they have undesirable and unpredictable properties. First, recombinant DNA technology was only used to clone DNA into the bacterial host, and we obtain the overexpression of the gene products. Now, genetic modified organisms can be constructed through recombinant DNA technology. It can be a transgenic animal or it can be a transgenic plant. Then, recombinant technology has a marvelous role in the field of medicine. We can imagine it from gene therapy, in which we can insert a normal gene by deleting the abnormal gene and can treat the genetic diseases. Now, in recombinant DNA technology, expression system is very much important. In expression system, we make use of two things. One is the host and other is the vector. The most commonly used vector is PBR322, but now there are different derivatives of PBR322, and one of the example is of PUC18, which is routinely used in the labs. Why is it so? Because it is completely sequenced. Now PUC18 is used in combination with E. coli K12 because E. coli K12 is non-pathogenic. It occurs in the gut of human beings and of animals. So here in the expression system we make use of biosafety level 1. Now sometimes biosafety considerations are required. For example, the DNA sequence that has inserted into the organism, it is actually obtained from the pathogenic strains. So here what happens, there is increased virulence of the transgenic organisms. So here biosafety consideration is required. The second thing where biosafety consideration is required, when the inserted DNA sequences are not well characterized, or when the gene products code for the toxins, for the cytokines, for the hormones, or for the pharmacological activities. In recombinant DNA technology, we commonly make use of viral vectors, and the most commonly used viral vector are the adenoviruses. Even for gene therapy, we make use of viral vectors. But one problem with adenovirus is that they lack replication genes. So the stock of adenovirus can get contaminated with the replication competent viruses. So here we have to look at the biosafety level. We have to treat the competent viruses at the same biosafety level at which the parent adenoviruses is treatable. Now, genetically modified organisms are very much popular nowadays, whether it is an animal or it is a plant. Now, mice has been constructed that express human poliovirus, but it has been observed that these transgenic mice can transfer human poliomyelitis. When histological sections of such mice was performed, it was found that the mice poliomyelitis is similar to human poliomyelitis. So such transgenic mice are dangerous. In recombinant technology, we construct new lines every day, but here transgenic animal studies are required, that what is their route of transmission, what is the inoculum size of infection, and what is the extent of virus shedding. So all these parameters should be recorded whenever we are dealing with the new lines of transgenic organisms. Apart from animals, we also make use of transgenic plants. For example, transgenic plants are nowadays resistant to herbicides, insecticides, and even to the antibiotics. So these transgenic plants are also very useful if we look at from the nutritional product point of view or from the medicinal point of view. But here risk assessment is very necessary. Inserted gene must have the known properties, that whether the gene code for the toxins, that whether we have that the gene which we have inserted code for the cytokines or they code for the hormones, and sometimes transgenic plants are also allergic when we are dealing with the nutritional products. So it is very important to note that whether the inserted gene has a code for any sort of allergens or not. Then gene expression regulators, enhancers, we have the information about them in order to sequence them when we are looking for any sort of mutation. Sometimes what happens in the transgenic organisms, proto-oncogenes get mutated into oncogenes and they become cancerous. So again, here risk assessment is required. It is also required for antibiotic resistance purposes, that if we are dealing with the expression system, then which type of plasmid we have to use as a vector.